So, how come spike protein can persist for so long post infection or vaccination? That's one of the biggest mysteries we had to contend with in this pandemic, and this will be the topic of today's video. Specifically, why could spike protein post infection last for so long? So, recently I made a video on the topic of how long spike protein can exist post vaccination, and we talked about that up to 6 months the spike protein can persist after vaccination, and then spike protein post infection can also last in the body for many months as well. My name is Dr. Mikola Rashik of Meragenomics. Let's get going and let me take you on a nice walk along with this conversation. And specifically, to summarize, the topic of today's video is the concept that the virus, SARS-CoV-2 virus, could infect bacteria so that prokaryotic cells, so specifically bacteria, could act as a reservoir for maintaining the virus. So, and this could explain, for example, as to why we could be detecting spike protein for so long. And I'll show you the evidence as well from the publication. This has been brought to my attention actually a long time ago already by one of the members of one of the long COVID associations from the UK. And they were telling me, listen, this is something we need to be looking into it. And that person brought a lot of the scientific information to my attention, but I haven't had a chance to really get to it in terms of video content till now. And here we are. We're going to start discussing it today. So, the paper I'm going to be talking about, the authors mentioned that, look, their own work previously have shown that they were able to grow the virus, SARS-CoV-2 virus, in bacterial cultures for up to 30 days. So they've already demonstrated evidence for that. They also brought up information of other people's works, and an example of that would be how when you get infected, the microbiome can change in an individual that is infected, and the pattern can be seen that the beneficial bacteria can decrease in their amounts, and the bacteria you don't want that will not be good for you, they can increase. So that has been presented before as well, and we did a video on microbiome post-infection as well. And they even brought up an interesting study discussing how computational modeling was done, showing that bacteria can produce compounds that will mimic ACE2 receptor, and therefore these compounds can start interacting with, say, spike protein, and therefore prevent the virus from being able to interact with our own ACE2 receptors on our own cells. And the computational modeling, that's also called in silico studies, meaning it's not real, obviously, it's computer modeling, but that was done. And the same authors whose work I'm discussing today, they've also, that's another evidence they've shown, is that when you culture the virus, SARS-CoV-2 virus, in the bacteria can start producing certain toxins. These are called endotoxins. Endotoxins, and some of these toxins, they mimic toxins that you can find in venoms of different animals. So that's interesting, right? And you might have been hearing how infected individuals, they can have these venom-like compounds in them, And these authors claim, well, these are produced by the bacteria as a consequence of infection. It's the reaction of the bacteria to the infection. And we'll discuss this a little bit more later. I think, if I don't forget, we'll see what happens. So there is an evidence suggesting that, look, bacteria very well could be inside our bodies, could very well be interacting with the virus, including the virus infecting the bacteria. So what these authors wanted to do is, of course, to prove it. And how do you monitor presence of a virus in a culture, in a tissue, whatever? There's few ways. And I wanted to break this down because this is very common question that I have. It's like, how do you prove that virus even exists? So remember, viruses, they require a host in order to replicate. So they need to infect some cells. Cells will then replicate the virus, and the virus can then come out of those cells. Okay, right? So how do you monitor that? So you can use microscopy, specifically electron microscopy, and we discussed this technology before, so check it out for background information. That's probably the closest we can get to taking actual photographs of viruses. You can also do fluorescent microscopy. This is very powerful technique. This is where, so you can do that. You can look at the proteins themselves of the virus. So one of the ways of looking at the proteins of the virus is fluorescent microscopy. The way that works is you target the protein of interest that you want with a specific antibody that will recognize the protein you want to see, and that antibody has a fluorescent molecule attached to it. So as a consequence of that, you can see the presence of the protein, viral protein, because you see fluorescence, right, under a microscope. So there's that. Another way of looking at the proteins, viral proteins directly, is a concept called mass spectrometry. This is basically where you take the proteins, you can break them down apart, cut them up, and then you see what fragments you produce, and you can get very precise information as to what was the content of these proteins. This technique has been around for very long time, almost a century, not quite, but long time. So this is one of the most precise techniques we have. And of course, there's other ways. One of the most powerful ones at the moment, thanks to the development of new technologies to decode genetic code, next generation sequencing, you can look at the genetic information of the virus. This is so powerful right now that we currently track virus evolution in exquisite detail. A lot of people don't appreciate how much information we're gaining just from sequencing the genetic information of the virus. And we're doing this all over the world. And we have exact information on the virus, down to exactly what is mutating in the genetic content. So you can track evolution of the virus in great detail. And then the last one that I know of, there might be more techniques, but these are the major ones, is you can also track how infectious the virus is by looking at how well the virus, for example, kills the cells that it's infecting, right? So there's multiple methods that you can use. And these authors, they use electron microscopy, and they basically showed, there's two methods they showed within electron microscopy. So they showed that when they co-cultured the bacteria with the virus, they were able to use electron microscopy to show that there's these round structures present within the bacteria, and in indicating or insinuating that these are very likely, obviously, SARS-CoV-2 virus, right? And that's, like I said, this is the closest we can get to actual getting photograph of a virus. So keep that in mind. And then to further corroborate that this is indeed the virus, they did another really cool technique that I also discussed before, where they took antibodies, but this time the antibodies to target specific virus protein, and they were targeting nucleocapsid protein. So that's the protein that's found inside the virus. It's not on the surface of the virus, but it's inside the virus. And the antibodies were targeting that protein, but instead of having a fluorescent tag on it, fluorescent molecule, instead it had a gold particle, super, super tiny, minuscule gold particle. And with electron microscopy, the way it works is you shine beam of electrons and they bounce off of three-dimensional surface. So they bounce off gold particle and you see a little black dot. So, and that clearly indicates where that viral protein is, and they were able to show, look, there are viral proteins inside bacteria. And in this image, they indicated the presence of the virus with those gold particles by pointing to it with gold arrows. And then they also showed, look, some of these bacteria are even being lysed, meaning they're breaking down, and basically that means the virus will be released. So they also showed that in those images. So in essence, this looks like a direct proof of the fact that bacteria can be hosts for the virus. So in other words, the SARS-CoV-2 virus, coronavirus, has a bacteriophagic behavior. What does that mean? Bacteriophages are viruses that infect bacteria, but coronavirus, like SARS-CoV-2 virus, doesn't infect bacteria. It infects our cells, human cells, or like mammalian cells, right? But clearly this also means that they can also infect bacteria. So this is really important, because what does this mean? It clearly suggests that the virus itself doesn't just have a respiratory component. There is a oral fecal route with the involvement of this virus. And the authors further discuss this by saying, look, this is corroborated by other type of evidence. For example, typically when people get infected, almost always you also see, when you test for it, you see the presence of the virus in the fecal sample and in the stool sample, or in other words, in your poop. So, and that could be potentially significant, right? And in this paper, the authors also said, look, my understanding is, because this is not my area, the way our authors were talking about this, they were mentioning, like, we're not really testing these viruses for their ability to infect bacteria, because that's, at least was my understanding, that we really don't have a good knowledge of how well these RNA viruses, whether they infect bacteria or not, and we should be testing for this. And they brought up a couple other examples. This is the interesting part. That was the interesting part for me. They brought up an example of HIV and poliovirus. So they mentioned that, look, there is evidence suggesting that HIV, which is another RNA virus like SARS-CoV-2 virus, could be infecting bacteria, because there was a publication indicating that bacterial samples isolated from a certain individual, I think it was a kid, from their throat and mouth area, those bacterial samples had a HIV genetic content present inside, right? So indicating that you might have a HIV infection. But the one that I found was more interesting was presence of a poliovirus. Discussion of a poliovirus. And the reason why I found this interesting is because poliovirus is another example that was believed to be only a respiratory virus at the beginning, and later on it was turned out to that it also acts via the oral fecal route. And one way that was demonstrated is that the poliovirus was capable of replicating inside our intestines, so in our gut, And when you provided oral vaccine against the poliovirus, that had the capacity to prevent that propagation of the virus inside the gut, but injectable vaccine where you injected in the arm, that did not. So the oral vaccine was attenuated virus, poliovirus, so that was effective against stopping the oral fecal route transmission versus the injectable route was not. And this is how finally scientists were able to confirm back then that this is not just a respiratory virus, but it has another route of transmission as well. And what was also interesting that, of course, poliovirus is almost eradicated completely at this point. There's only a couple countries where it's still endemic. We nearly eradicated this virus with vaccines from existence in the world, just like we've already achieved for smallpox. And unfortunately, polio is popping up here and there, luckily in very, very small quantity, meaning very few individuals are infected here and there, but it's still showing up. So the goal is still to eradicate poliovirus completely. And so we have decades and decades of knowledge and information on this virus. And scientists have previously shown that the bacteria, when they interact with the poliovirus, can also influence how the virus mutates as well, and it can achieve that by releasing, or how the virus propagates, I should say, and it can achieve that by producing certain endotoxins as well. So bacteria can manipulate the virus, and the bacteria can protect themselves against the virus as well, which, remember, the authors showed, whose paper I'm discussing right now, they also have shown that apparently in the past that bacteria can produce these endotoxins when encountering SARS-CoV-2 virus as well, these venom-like toxins, right? So the authors proposed this really interesting question is, could we produce vaccines that are probiotics by basically influencing our bacteria in order to be able to fight viral infections? So that brings me to now another paper of these individuals, and where they discuss this concept in a little bit more detail. In this paper, they showed more of the same evidence. So once again, they showed electron microscopy images showing how bacteria culture was again infected by the virus. You could see those round structures. Again, that's as close as you're gonna get to seeing viral particles. In this case, inside bacteria, no less, as opposed to outside. And again, they use the immunolabeling, where they use those amino acids to again target nucleocapsid protein of the SARS-CoV-2 virus, to show, with the gold particle attached to it, to show that you can see presence of those gold particles right inside the bacteria. So again, it's more of the same evidence. But they also discussed how bacteria, because of course bacteria, remember, they have their own machinery to produce things. They can chemically change amino acids. Amino acids are the building blocks of proteins. And there's about 20 of them. And basically all your proteins inside all the cells inside our body, they're made out of these different building blocks called amino acids. And proteins are, of course, the machines that do pretty much almost all the work inside our bodies. There are other genetic content can also have a machinery-like behavior, but it's somewhat primitive in comparison to what proteins can do. Proteins are genuinely molecular robots. They can do amazing things, and spike protein is an incredible example. Excuse me, of that. So check out my video on that topic, what kind of a robot spike protein is and what it can do in order to actually ensure that the virus can infect the cell, right? So because bacteria are are different and they have different machineries inside them, they can alter the amino acids. And these altered amino acids, these authors refer to as D amino acids. And you can track these amino acids. And if you detect their presence, it means their origins are of bacterial origin. So, and what these authors mention is that there's evidence that the spike protein isolated from patients who were infected with SARS-CoV-2 virus, they have presence of these D amino acids, meaning of bacterial origin. So that's another way of actually monitoring the possibility that that bacteria are infected and therefore the spike and produce progeny of the virus and therefore can act as this reservoir. So that I thought was also quite interesting. So there's another way of how that information can be tracked. So there is stacking evidence suggesting that the bacteria in our body could act as a reservoir for the virus, so that a person could be infected, you can clear all your symptoms, and you no longer will test positive on the test from your nasopharyngeal sample, meaning from your mouth or throat. You will test negative, so it seems like you no longer are infected, but you could still test positive on your fecal sample for the virus. And the authors even mention as well, they've seen that in their own work as well. They've presented that in prior publication that has been seen with in the work of others as well. And so pretty, the sunrise. And therefore, when people are, for example, presented to the hospital, it's been suggested that once you stop testing positive for your mouth sample, you still should be potentially testing the stool or poop sample, especially if a person has a tendency to show frequent infection. So very, very interesting stuff. But here's the fun part, right? Because it clearly, if there is an oral fecal route for the SARS-CoV-2 virus infection, that also changes how we should really be targeting this virus and what we should be doing for treating people who are infected. It just changes epidemiology of how we interact with the virus, right? So we need to basically consider this, and that gives new opportunities. And they brought up the concept, which I thought was interesting, of maybe developing probiotic vaccines. So what does that mean? Look, we all know that if you take viruses and you expose, if you take bacteria, not viruses, and you expose them to antibiotics over and over and over, the bacteria will have the capacity to mutate because it can replicate fast and eventually build antibiotic resistance. That's one of our big problems that we now have in medicine, is that many of the bacteria are becoming resistant to many of our antibiotics. Well, then the question is, could the same happen when bacteria encounter virus, which is of course attacking the bacteria and destroying it? So then when you have a repeated exposure to the virus, can then bacteria build resistance to the virus? And there is suggestions that it can. And one of the ways that bacteria can achieve that is by mutating in order to produce these specific compounds, endotoxins, that are used by the bacteria in order to fight the viral infection. And so could we take advantage of that? And could we, for example, culture the bacteria with the virus to create bacteria that are resistant to viral infection, and use such bacteria subsequently as a probiotic vaccine, because that would basically mean that we could seed bacteria that would be resistant to infection by the virus, and therefore we could prevent such reservoir capacity of the virus, and therefore be able to treat the infection even better than what we are just doing right now, which at the moment is pretty much all we're doing at the moment for SARS-CoV-2. The only approved method is, as far as I understand, is the vaccines, right? We don't really have yet any approved treatments for COVID-19. So very interesting concept, and I will be bringing up in other videos other concept of how we could be developing another forms of vaccines as well. This is just one really interesting idea that I wanted to share with you. And remember, bacteria is not the only source of cells in our body that could act as a host of reservoir for the virus. Another one, of course, I was telling you that neuronal cells can be infected as well. And recently there was a publication that I'll discuss in another video, that was shown in monkeys that I believe it was macrophages, can act as a reservoir for a very long time as well, despite the fact that there is no detectable SARS-CoV-2 infection, not via the traditional methods of testing for it, while the reservoir can exist for many, many months. So there's multiple cells, for example, possibly multiple cell lines in our body besides the bacteria themselves that could act as a reservoir. Oh, and one more thing when it comes to the bacteria. One of the authors, these authors who were showing that the bacteria are being infected, they also brought up a work of other individuals who also were showing this. But here's the interesting part: this other scientists also showed that certain antibiotics, when administered to the bacterial culture that were propagating SARS-CoV-2, were capable of reducing the amount of virus. And this is why you might have heard the possibility of maybe using antibiotics to deal with long COVID. Now, this is not an approved treatment at all, but you might have heard about this. And this is why some people think, hey, listen, if our gut is affected by the virus and you're having long COVID because of that, because there is a reservoir of virus in your body because of the bacteria, one way possibly to treat that is via antibiotics. But obviously we need proper investigation, proper clinical trials to really determine that, okay? And we don't have that available at the moment. But if you've heard about it, this might be the reason why, okay? All right. Thanks for joining me for this wonderful walk. Thanks for hanging out with me. Thank you for your support. Hey, thanks. Please give us a like. Please share the video. This is how we grow. This is important for us. And I look forward to seeing you in another installment. And as always, please please go outdoors and enjoy yourself in Mother Nature. All right. That's it from me today. And you guys have an amazing day. Bye, everyone.